This is the effect of indirubin E804 as an alternative treatment to rheumatoid arthritis and Danielrhea. Zebrafish was discovered as a model organism in 1960 as a tropical fish native to Southeast Asia. As embryos, they are fully transparent, and as they develop, black stripes begin to appear and give them a blue tint. After fertilization, zebrafish eggs develop outside of the mother's body so they can be studied early development during experimentation. They reproduce at an extremely fast rate, producing hundreds of fertilized eggs weekly. Zebrafish share 70% gene homology with humans in general and precisely 84% regarding diseases, making them viable for research over hereditary diseases and causes that cannot be tested in humans normally. Embryos do not develop spinal cords till after 7 days post-fertilization, which makes them fantastic models for invertebrate research during that period. In 2001, the Sanger Institute began the Zebrafish Genomic Sequencing Project. The entire sequence is almost complete, still being updated today. 14,000 genes have been mutated to study their function. Tissue inflammation is a chronic issue across the body that ties into multiple diseases, including rheumatoid arthritis, or RA. Inflammation occurs as a response to eliminate an unwanted infection or injury, and this is mainly controlled by the cytokine interleukin-1, IL-1. IL-1 creates an inflammatory response controlled by the immune system to illness or injury, allowing it to control unwanted factors before it creates mayhem. IL-1 is secreted by the monocytic cell in the extracellular environment that creates a series of microvesicles connected from the inside of the cell to the plasma membrane. This series is mostly unidentified and not adequately understood, as there have not been many attempts to characterize the pathway itself. As zebrafish research has continued, they are instrumental to where targeted responses by IL-1 have appeared with little to no cause, initiating chronic diseases like RA. RA is a chronic autoimmune and inflammatory disease characterized by swelling in certain affected areas of the body. The immune system attacks the healthy cells in the body without reason, causing swelling of the tissue and joints in the hands, knees, wrists, and ankles. This persistent inflammation decays the joints over time, causing chronic pain, inability to maintain balance, and visible deformity of the joints around the body. RA may affect certain parts of the body, such as the lungs with asthmatic symptoms, hearts with premature chronic disease, and pancreas with diabetes, as collateral to their attacks throughout the body. This disease has no specific cause, but certain factors are connected to this disease, including age, sex, and genetics at times. Rheumatologists specialize in treatment of this disease if ever diagnosed. RA cannot be diagnosed by a single test, but by a collection of observations, x-rays, and tests along with a familial history chart. Once diagnosed, RA is treated with inflammatory suppressants, which have been known to leave toxic effects to the body. This usually has two types of treatments: disease-modifying anti-rheumatic drugs, DMARDs, or biological modifiers that can be used to relieve pain and increase joint function. These are medications that block the immune system and prevent inflammation signaling by IL-1. They sometimes are combined with other drugs and are usually not effective for a minimum of 6 months after beginning use. Examples of these medications are Aleve, which is known to cause low blood cells, neuropathy, gastrointestinal or liver problems, and high blood pressure. Biological response modifiers such as Actemra are more specific type of these medications that target the immune system to treat the core of the problem, yet they have lasting effects. Moreover, the patient is more susceptible to severe illnesses such as tuberculosis, upper respiratory infections, or bacterial or fungal illnesses. But lately, homeopathic treatments have severely decreased symptoms and relieved most of the inflammation without toxins. Indirubin E804 is an extract of the indigo plant used medicinally to treat inflammatory diseases because of its numerous anti-inflammatory and anti-angiogenic properties. Studies between human diseases and indirubin are limited because of its recent introduction into the scientific community, so far showing that it can efficiently decrease inflammation without leaving lingering chemicals or toxins in the body. It has been used as a natural therapeutic in chemotherapy, where it has also been successful in shrinking tumors and preventing them from metastasizing. Indirubin targets inflammatory receptors such as IL-1, cyclin-dependent kinases or CDKs, and STAT signaling. There have only been a few cases where the plant has had an adverse effect, all of which can be caused by mistreatment of gastrointestinal issues instead of anti-inflammatory ailments. The purpose of this experiment is to determine whether a natural therapeutic can serve as a primary treatment for biologically modifying diseases, and the variables are the independent is the treatment and the dependent is how the embryos respond. The hypothesis is that if inflammation-induced zebrafish models are given indirubin to monitor and treat tissue inflammation, then the indirubin will have a higher efficiency in decreasing inflammation and decreasing kidney toxicity from generic medication and preventing symptoms that correlate to rheumatoid arthritis from occurring. The basic materials are listed on the slide, such as the wild-type zebrafish embryos received from the University of Miami and the indirubin E804, along with GFP assay kits and rheumatoid arthritis assay kits with trican methanol sulfate. The procedure began with the maintenance of the zebrafish, along with receiving them from the core facility and then transferring them into 10 petri dishes, depending on the variable, along with taking pictures with the microscope every day using a camera. And then 2 assays were applied. The toxicity assay tested for phenotypic tests and samples collected to be measured for chemical level in the systems, and the mortality assay was tested on the first and final day to count for the amount of living zebrafish embryos at the end of the project. The last assay was the rheumatoid arthritis assay, which used the leukocyte migration testing to test whether at the site of an injury, the leukocytes would migrate at a quicker rate depending if they were exposed to the medications or not. After an extensive data collection period, the results were evident in proving one point. The hypothesis was only partially supported. The original hypothesis stated that the indirubin E804 would function directly as a primary medication without the support of toxic generic NSAIDs such as Aleve. At the end of the experiment and looking back towards the properties of indigo itself, it was possible to conclude that the filtrate properties had a significant impact on the potency of indirubin. While having an efficient support towards the Aleve already present in the system, it is visible that the E804 worked in lessening the inflammation and filtering out foreign toxins and working to extend the lifespan and lower the toxicity within the system. Data from the toxicity assay presented the Aleve itself had a 77.8% toxicity level, higher than the normal compared to the 33.4 created by the E804 and the 22% generated by the combinational test. In efforts to provide backup to the NSAIDs, the indirubin not only created an accelerated rate of cellular movement towards the site of injury, but also managed to decrease the STATs and IL-1 signaling to lower the inflammation block itself created by the event of stress that chronic rheumatoid arthritis causes. Copper sulfate models were used as a control or baseline to compare against the treated variables, which allowed for certain aspects of the data to be reevaluated for discrepancies that went unnoticed. It is possible to derive that the E804 buffered the Aleve to decrease IL-1 signal boosting and overpass the inflammatory barrier while also improving the leukocytic transportation towards the site of injury. The experimental results have proved that while NSAIDs like Aleve are designed to treat primary medications, they are in fact not the most beneficial when it comes to maintaining the chemical balance. Therefore, natural therapeutics have benevolent impact towards treating inflammatory diseases such as rheumatoid arthritis. Most importantly, derivatives such as E804 can be part of a regimen that works with secondary buffers to improve the efficiency while also decreasing the toxic chemical exposure common in treatments to help without harming. Research towards diseases such as inflammatory and arthritic diseases is extremely sought over in the medical community because of the great amount of people affected. Expanding current projects such as these to include other experimental medications as primary and secondary additives to regimens to lessen the side effects and more efficiently treat such problems is astoundingly beneficial. In addition, research with mutant lines of model organisms to portray the victims of these illnesses would work in favor towards the accuracy of the data and applicability in general towards the public. The data of this experiment could be applied to real-world situations, including the widespread tissue inflammation caused by arthritic diseases, which affects about 54 million adults in the United States. Research over the years has focused itself on discovering a primary medication and has been successful to some degree. Although the research has resulted in numerous treatments, scientists continue to look for less toxic alternatives. Throughout this experiment, a few obstacles were encountered. At the beginning of the experiment, GFP mutants were available to collect baseline data for the rest of the investigation. Still, unfortunately, due to COVID restrictions, they were no longer able to be used as model organisms. Similar models using copper sulfate as inflammatory agents function to stimulate the mutant effect, allowing for baseline data to still be applicable during the experiment. Furthermore, the models introduced to copper sulfate have been adversely affected compared to the models of zebrafish specifically exposed to medication without inflammatory agents. Thank you.